Hi, my name is NAZATUL ANISHA. So, histology is the study of the microscopic anatomy of cells and tissues of biological samples by observing the prepared slides under microscope. So, in the field of histopathology, there are 8 important stages in preparing the histological slides, mainly for research purposes. So, each step has its own standard operating procedure that must be followed accordingly in order to produce a high-quality histological slides, which contains tissue that was properly sampled, fixed, embedded, microtome, and stained without artifact. So the aims of this final project is to help the students gain more experiences and knowledges, particularly in histopathological processes. Next, to familiarize the students with handling the instruments and materials in a proper way. Last but not least, to hone the students' skill by performing every process individually.
Hi, my name is THIVASHINIE. The first ever step in making histology slide is specimen logging. In specimen logging, the request form will be checked for the patient's name, date of birth, sex, address, a medical record number, locations such as ward, clinic, and emergency room. Relevant clinical details and physician signature must be checked. Number of specimen taken based on the container or number of container and source of the specimen such as biopsy site together with the date of specimen also will be checked. Not only that, but the specimen container will also be checked for some details such as the specimen of the container is properly filled, patient's name and medical record number, source of specimen and the specimen number. The apparatus used for our preparation is labeled specimen and specimen logging form. The image in the result shows the result we gain, which is the mice specimen logging form.
My name is Felicia and I'll be explaining about step 2, which is dissection of mice, extraction of organ, tissue grossing and fixation. For dissection, latex gloves were used when handling the rat and safety goggles were required to protect the eye from chemical spattering or debris. Next, the rat was placed in a dissecting tray and external features were examined. Then, cervical dislocation was performed to euthanize the mice. The skin of the rat was removed to expose the muscles. The skin was lifted and snipped away from the muscles using scissors and forceps. The abdominal wall was cut through in the mice. Once the body cavity was opened, it was rinsed in the sink. The target organ was removed by cutting the attachment to it. The organ was rinsed in the sink with saline water and submerged informally in a universal bottle. Next, for tissue grossing, the specimen was inspected with the bare eye to obtain diagnostic information while being processed for further microscopic examinations. And lastly, for fixation, the specimen was cut into a suitable size. The specimen was fixed with 10% formalin.
The formalin-fixed tissue was immersed in a solution for the tissue processing according to the time as shown in the table below. For the tissue processing, consists of 3 steps, which is dehydration, clearing, and infiltrations. For dehydration, we remove all the water content in the tissue sample by immerse the gradient of alcohol, which is 50% of alcohol, 70% of alcohol, 90%, 95% of alcohol, and 100% of alcohol in order to remove all water content in tissue sample. Next is a clearing. Clearing by using the xylene, which is an intermediate agent for clearing all the alcohol that contain in our tissue sample. After clearing all the tissue sample with the alcohol, now the tissue sample will be introduced to the molten paraffin, which is we call it infiltrations, which is the specimen will stay overnight in the molten paraffins. As a result, this is our sample before tissue processing, and this is our sample after tissue processing.
The next step will be tissue embedding and blocking. So embedding is the process where to enclose and to support the specimen in order to produce the tissue block. So firstly, the lid of tissue cassette was removed to expose the sample tissue. Then the lid was used to place the tissue on and bring it to the embedding machine. The mold was placed on the paraffin dispenser unit and filled with a small amount of paraffin wax. Next, the sample tissue was taken from the lid by using forceps and placed at the center in the mold. The mold was then moved to the cold plate to allow the wax to solidify for a few seconds. The mold was then moved back to the paraffin dispenser unit and covered with the lid once the mold moved to the cold plate. After placing the lid, more paraffin was dispensed to fill the mold until it overflowed. The mold was then quickly moved back to the cold plate to allow the wax to settle for a while. Last but not least, the mold was finally transferred to the cooling unit. So these are all the instruments and materials that you will use in embedding process, which are paraffin dispenser unit, cooling unit, paraffin, cassettes, metal mold, and forceps. That's all from me. Thank you.
After embedding, the step sectioning using microtome fishing and mounting was performed. The apparatus used for this process is a rotary microtome, which is responsible to section the paraffin block into thin section, then flotation bath, which will flatten and unwrinkle the sectioned ribbon. The method is the block was inserted into block holder in the microtome. The block was moved forward until the tissue surface was almost touching the knife edge. The surface of the tissue block was pared off to expose the tissue face before taking sections. The microtome gauge was set for the desired thickness. The microtome was operated until complete sections were being cut and maintained a cutting rhythm. The section allowed to be floated in the water. The section was picked up from the bath by immersing the slide. The slide was placed on the hot plate to dry before staining. The image in the result shows the histology slides after complete sectioning and fishing. The bottom figure shows detailed flow of this process with captions.
Hello, my name is SYAFIYAH NAJIHAH. So H&E stain is used for the staining process. All the solutions that we use and the time for each tissue staining is according to the workflow as shown here. These are all the instruments that are needed, and these are the results where the hematoxylin will stain the nucleic acid, which appears as purplish blue color, and the eosin will stain cytoplasm, which has a pink or orange color. Next, the stained tissue cell was dried and protected by the PX mounting method in order to preserve the stain and is ready for observation. The steps are shown here, and these are all the instruments that are needed.
My name is Mohamed SULAIMAN, and I will be explaining for step 7 and step 8, starting off with microscope slide viewing and labeling. So the slides were observed at 2 different points, one after hematoxylin, and here we can see that the slides have been stained in a light pink. This is because only the nucleus has been stained at this point in a dark blue color. And the second time observation was after eosin, and the specimen looked much darker stained here because eosin has stained in a reddish pink color to the cytoplasmic components. And the observation used was 4 times objective lens in a light compound microscope. And this is the evidence of the slides air drying after DPX mounting. For the labeling, however, of the self-obtained slides, this is the liver slide, and this is the kidney slide, and this is the brain. And the comparison microscope was used to observe some of the other slides actually. And this is the kidney at 20 times, this is the kidney at 10 times, and also label. And this is the same using the comparison microscope, and this was the heart at 10, and this was the heart at 40 times magnification. So finally, for recording data and reporting, we can see these are some of the artifacts, and the right is actually the properly stained hematoxylin, and on the left, it is actually expired hematoxylin, which couldn't do the staining properly. More reports include that the self-obtained cells, although they were not the perfect type of specimens, they were good enough for observing structure and morphology, while for the comparison microscope observed once, they actually allowed a very high magnification for 100% like accurate observation and all the perfect morphology and structures down to the cellular level. That's all for me. Thank you.